Good. So, good evening, ladies and gentlemen. Thanks all for coming out on this evening when there's a lot of competition with Albert Schwarzenegger speaking at the Republican convention tonight. So, I congratulate you on your choice. Tonight we have a great lecture. This is Graham George, who is in fact a defector from SLAC. He was with us for 11 years at our synchrotron radiation laboratory here, and then was lured away mysteriously to Canada, where it's cold and wet all the time, rather than California. So I'm not quite sure why he did that. There must have been a good reason for that. Tonight, Graham's going to talk to us, I believe, about metal and fish. I spent a lot of my time, wasted a lot of my time trying to get metal into fish in the form of hooks without much success. So I hope I may get some hints tonight. So let's have a big round of applause for Graham. Thank you.
Well, thank you very much, Neil. Is this on?
Yes.
Yeah, okay. Tonight, what I'd like to tell you about, well, the title of my talk is Metals, Molecules, Life and Death. I want to tell you about metals, how they crop up in molecules, and how these are important in our lives, and how they can sometimes cause death. Hello? Oh. So what is a metal? Metals are very familiar. They're all around us. This is a picture of some sheet stainless steel, a copper kettle, a Coke can, a copper mug, and some screws, and we're very familiar with them in our everyday lives. If you look very closely at a metal, then you'll see, you can actually see that pure metals are composed of one kind of atom. This is a sort of a chemist-style rendition. These little balls I'm going to be showing you a fair number of are all atoms, and the little lines between them are where they're joined to each other with what's called chemical bonds. That means they're just attached to each other. And as I said, pure metals are composed of just one kind of atom, and so this is what you might look if you looked with a very powerful structural tool at some of the metal in this copper mug. You'd see a whole big bunch of individual copper atoms all bound to each other. They're all around us, and they occur not only in pure metallic form, but also in molecules. Now, I'm going to come back and I'll tell you what molecules are in a moment, but first I'm going to tell you some sources of metals in our environment. And you can crudely divide them up. One source of metals is natural sources. They occur in our environment through weathering of rocks and minerals, and they'll be dissolved, for instance, in natural waters. They're emitted by things like volcanoes. Exotic metals in forms such as iridium pentafluoride come out of the top of volcanoes. They also occur from man-made sources, and there's a lot of interest in man-made sources of metal these days. Things like farming, particularly organic farming these days. Metals are becoming very important in that. Other man-made sources include mining operations, where things like arsenic are released into the environment in incredible quantities sometimes. So what are the sources of human exposure to metals? Well, mainly from our food and also from our drinking water. And these are usually beneficial, and sometimes they're not. We'll talk about that later. These are usually beneficial sources. Some metals we really need, and I'll say more about that. So food and drinking water, and often the bad sort of metal exposure is direct contact. You can see someone here doing something unwise with a little bit of mercury. So metals in humans. They have two basic roles they can fulfill. One is essential metals. So we need metals to stay alive. They're very important to us. Most of us, or at some time or other, have taken vitamin or mineral supplements. And you'll often see such things as copper in there, certainly iron, maybe zinc, molybdenum, and selenium. Selenium's, we know increasingly that selenium is very important. And I'll be telling you more about selenium. Now, I'm going to say at this point that I've been pretty generous in my definition of a metal. Selenium's actually not really a metal. It's something called a metalloid. But metals, molecules, life and death sounded a lot better than metals, metalloids, molecules, life and death. So I've been a little bit generous. And that's really for any chemists who are in the audience. So I'm counting things like arsenic and selenium as metals for the purposes of this talk. Then there are other metals. Some of these have toxic effects, and here are some well-known ones that do. Arsenic, mercury, lead, and cadmium. These have no known role in our bodies. Arsenic may have. At the end, I'll answer questions, but I can tell people about what roles arsenic might have at the end if they want to know, but I'd prefer you didn't ask questions during the talk, but save them for the end, please. Mercury has no known role at all in biology. Cadmium may be very important, it turns out, in controlling global carbon dioxide, how much is dissolved in the ocean. I'm not really going to say anything about that, but it's not important for us, but it may be very important in other organisms. And just about any metal is toxic at a high level. So if you take enough of it, it'll do you harm. So, as I mentioned earlier, this is a flashback to a previous slide. Metals are all around us. They occur in metallic form, but also in molecules. And these are the things I really want to talk about, are the molecules. So what's a molecule? This is a picture of a molecule. And again, the little round balls are atoms. And this in the middle is a copper metal atom. And in this case, if you remember that previous picture I showed, we had copper bound to a whole load of other coppers. In this case, we have copper bound to 3 of these yellow atoms, and these are sulfurs. Now, it doesn't really matter what they all are. The point is this is a discrete entity. It's a discrete little unit, a little packet of material, if you like, in which different atoms are arranged, and we call these things molecules. How big are molecules? Well, the length of the distance between the copper and sulfur is about 10 billionths of an inch. These are pretty small things, little tiny things, but they're very important, as you'll see. Another example of a molecule, a bigger one this time, occurs in us. Iron, which is a metal, as most of us know, is an absolutely essential component of blood. It occurs in a protein called hemoglobin, and here's a picture of it. This is a cartoon representation where many of the atoms are drawn in this very stylized ribbon. In fact, there's lots and lots of atoms all the way around, and if I drew every atom in the picture, it would look confusing to you. The important part I have drawn properly, and you can see in the middle, here is the atom of iron bound to 4 nitrogens in what we call a heme group, and it's very important. It's the thing that does the moving of oxygen around in our bodies. So this is an essential thing for us to stay alive. We need iron, absolutely. We can't live without it. And there are many, many thousands of ways in which we use different metals in our bodies. Now, what I want to concentrate on, actually, is one of the main things. This is really the main point of the talk, that molecular form matters. If I was to say arsenic to you, what would you think of? Poison, yeah, that's what everybody says. Everyone thinks of poison when they think of arsenic. And this is a famous movie, and it's really infamous as a poison since the Middle Ages. It's thought to be responsible for numerous deaths of important people, including Napoleon Bonaparte. It was possibly used by the Borgias in poisoning their dinner guests. And it's also a leading candidate for infant cot death, SIDS. I can tell you more about that at the end. I'm not going to mention it again in the talk. But molecules are important. And sometimes arsenic is poisonous, but sometimes it isn't. Here's a case where it is poisonous. This is lewisite, otherwise known as dew of death, the dew of death. It's a war gas. This is the forearm of a Marine who, in Iraq in 91, you can see this photo was taken in 1991 in May, was exposed to a small amount of lewisite that Saddam had at that time in his possession. He rolled up his protective suit and his forearm was exposed. And you can see for the short exposure to the small amount of lewisite caused all these nasty blisters. It's terrible stuff. It's deadly poison. If you ingest it, it'll kill you dead. So this is a very toxic arsenic molecule. The arsenic atom is this purple atom right here, the sort of shiny metal-looking one. The scientific name for it is chlorovinyl arsine dichloride, in case anyone cares. But there are other molecules, such as arsenobutane. Arsenobutane you get in fish and in all seafood, really. You get it in lobster, you get it in shrimp, you get it in crab. Any kind of seafood that you ate, you can have up to 0.02% in fish. And that's an awful lot. That's just tons of arsenic. But nobody ever worries about being poisoned from arsenic from eating fish. And you shouldn't worry about it either because it's non-toxic. This is an example of an arsenic molecule that's not toxic at all. We eat it, it enters our bodies, it circulates in our bloodstream. In due course, it's excreted in the urine. It doesn't harm us. It's not a toxic compound. And the difference is in the molecule. The molecule is very important. Same element. If anyone's a chemist, they're even the same oxidation state. I'm not going to define oxidation state because I don't want to bore you with details, but it's the different molecule, same element, different properties. Molecules are very important. Another example, everyone knows that mercury is one of the most toxic elements. It's almost common knowledge, but not in all its molecular forms. Cinnabar is a good example of a relatively non-toxic form of mercury. So here you can see a fragment of the structure, the molecular structure of cinnabar, and you can see the shiny atoms, those are the mercury atoms, and the yellow ones are actually sulfur, but that's actually unimportant to the point. But cinnabar has been used in jewelry, and here's a pendant made of cinnabar. People have constructed drinking cups of cinnabar and drunk from them regularly, and they've suffered no apparent short-term ill effects from this exposure. I wouldn't recommend drinking from cinnabar cups or actually wearing cinnabar jewelry, but you can contrast it with this substance, dimethylmercury, and a few drops of this on your skin will kill you. It looks just like water. It's a little sort of clear liquid, and you can see a little bit in this vial. This is dimethylmercury in a little bottle. A few drops on the skin will kill you. So clearly here the difference is in the molecule. Molecules matter. It's very important what the molecule is. So it's not just the metal that matters, it's also the molecule. And what molecule a metal is contained in affects absolutely every aspect of how it will affect us and our environment, whether it's beneficial, toxic, or benign. It's vital to know the molecular form if we are to understand these things. So now I'm going to move on a little bit and say, how can SLAC help? And this is a picture of SLAC site, excuse me. And many of you, especially if you've been to earlier lectures, will be familiar with this. This is the 280 freeway, this is the LINAC, and the part of SLAC I'm going to talk about is this part down here, which is SSRL, the Stanford Synchrotron Radiation Laboratory. I used to work there for 11 years, and I still come back there to do experiments. And Herman Winnick talked about, gave a lovely talk on April 27th. Maybe some of you are here. Synchrotron radiation, the light, fantastic. And here's the URL where you can find information, or actually watch the lecture online if you wish. And this is the Stanford Synchrotron Radiation Laboratory. And this can help us understand molecular form. It helps by being a source of what's called synchrotron radiation. This is a very, very intense beam of X-rays. And here you can see this is a sort of pipe. This is a little short movie clip that's been taken of a beam of X-rays passing through air. And you can see it's emerging from here, and there's a little mote of dust. It's repeating in a loop. A little mote of dust being caught in the beam here as it hits the lead beam stop over here. Now, this is like what would be called white radiation, so this is many wavelengths emerging, and very much brighter than that we'd actually use. But it makes the point, the air is actually glowing. There's so much radiation here. This is a very bright beam of X-rays. And we can use it in many ways. It has applications, as Herman pointed out, in physics, chemistry, biology, medicine, environmental science, geology, biochemistry, materials science. You could put almost any label around this. This is a picture of what a synchrotron, stylized picture of what a synchrotron might look like. But basically, you could put any label around here, any scientific field, and you'll find examples where synchrotron radiation has benefited us, and we know a lot more about some aspect of it than we would have done without it. It's a very important scientific tool. But why X-rays? This shows a length scale in meters along here. I didn't have the energy to convert it to inches for you, but I expect most of you know how big a meter is. And so this is a tenth of a meter, this is a hundredth of a meter, a thousandth, a 10,000th, a millionth, a billionth, and so on. And the property that makes X-rays useful to us is that they have a wavelength in the length scale of molecules, in the molecular range. So they are able to probe things in the distance range that we're interested in. How do we do this? Let's contrast it with conventional analysis. Let's say there's a metal in a plant, this plant for example, and we think there's this kind of a metal, it doesn't matter what it is. This is actually a molecule that we subsequently identified in this very plant, actually, down at SSRL, down the hill. But let's say we want to look at it with conventional chemical analysis. Well, basically, this involves usually chopping up the plant. You can see there's supposed to be little bits of the plant here floating in this conical flask. Chopping it up and usually dissolving it in something like acid. It would vary depending on what you're analyzing. And essentially, the molecular information can be wholly or partly lost with conventional analysis. So you end up not knowing about the molecule. But with the X-ray techniques given to us by synchrotron radiation, we have a direct probe. We can look directly at what's in the plant. In fact, you can even take the plant away afterwards and let it grow if it's a plant. It doesn't have to be destructive at all. This is a direct probe. The molecular form is intact. We'll know what's in it. We don't hurt this. We don't dissolve it. We don't change the molecular form in any way. It's what we call an in situ probe. It's a very neat way of understanding what molecules there are in complicated systems like tissues of plants. A couple of examples that I'm going to discuss. Here's mercury. I showed you a picture of someone doing something unwise, which was a modern picture. And this is something even more unwise. This is liquid mercury here. I did have a picture of a girl in a bathing suit taking a bath in a bath of mercury, but I've lost that. I couldn't show you that today. And that was from the 1950s or something, when people really didn't appreciate the dangers. Mercury is a very toxic element, but not in all its forms, if you remember that point. That's an important point. And there, unfortunately, are many tragic human-caused problems that we can find, including Minamata Bay in Japan, where dumping of industrial waste into the bay by Chizo Corporation caused large-scale poisoning of villagers in the nearby village of Minamata. And mercury poisoning has ever since, or certain types of mercury poisoning, has long been known by the name Minamata disease. This is in the 1950s. Later on, in the 1970s, Iraq, there was an incident in which ethyl mercury chloride dusted wheat, which was meant for seed stock for planting in fields. And it was dusted with this stuff to prevent fungi and pests from feeding on the wheat. It was used to bake bread instead. It was painted bright orange and it had a skull and crossbones painted on the sack. But that didn't mean anything in this sort of Iraqi culture. So people used it to make bread, unfortunately. And nearly 4,000 people died in the 1970s. And as you'll hear later, it's a pretty unpleasant death. In the Amazon basin right now, gold mining operations giving rise to a lot of industrial effluent, plus leaching due to deforestation, are causing widespread poisoning of mercury poisoning of the Amazonian natives. So mercury is and I could find lots more examples. There are many, many examples where mercury has been a really big problem. And unfortunately, we know a lot about its toxic properties. What about iatrogenic sources? I use this word because it's the sort of word your doctor might use. Iatros means physician in Greek, and genic means induced by, so this is induced by your physician. Sources from the health industry. One source that probably many of us, including me, have plenty of is silver mercury amalgam in the teeth. Silver mercury amalgam in teeth I have quite a lot of them. I had them from fairly early childhood. They're widely regarded to be relatively safe. And if they're silver fillings, they're silver mercury fillings. They're a mixture of silver mercury. And you can see here someone's teeth with a few fillings in them. A recent study in sheep using radio-labeled mercury showed that the mercury was in fact, this is a radioactive mercury isotope in the fillings. They drilled the sheep's teeth, they filled them with silver fillings, and they showed that the mercury was mobilized to the brain, kidney, and liver. Now, we don't know what the molecular form is, we don't know what it's doing there, but we suspect it's up to no good. Another source that's been in the papers lately is thimerosal in vaccines, and it's the subject of some controversy. I'm not going to comment on that directly in my talk. It's been implicated as a possible cause of some cases of what some people have called rapid onset autism. Fortunately, most childhood vaccines no longer have this mercury compound in them, so it's mostly eliminated. I think probably all these days. I think my three kids, my first kid who's 8, he had quite a bit of thimerosal. I didn't know, but wasn't thinking about it in those days. My second kid, I put a stop to it very early on, but he still had some from an early age. He's 5 now, and my 3-year-old has had none whatsoever. I'm going to be talking about things that are neurotoxic, so I better define what that means for those of you who don't know. When something is neurotoxic, it is a potent toxin, or it is poisonous to our nervous systems, including our brains and our peripheral nerves, depending on how it manifests its poison. That's what neurotoxic means. We're very worried, all of us who think about fish are very worried about how much mercury and how bad mercury in fish is for you. Fish are the primary source of mercury in our diets, and they are a major source, the major source of a neurotoxic methylmercury species. And this is well known. If you read the newspapers, you'll read about methylmercury in fish and how it's bad for you. How much mercury in your diet is safe? Well, for ages and ages, the EPA and the FDA disagreed on this. The FDA used to say, eat lots of fish, it's very good for you. And the EPA used to say, don't eat fish, it's very bad for you. Now, since March 2004, they issued an historic advisory. Here's the URL if you're interested. If anyone doesn't have time to write that down, come and see me and I'll get it off the laptop. And they said, eat no more than 2 fish meals a week. And do not eat fish that are high in mercury. Okay? Which fish are high in mercury? Well, they published a list. They're things like large predatory fish, fish that eat other fish, that are at the top of what's called the food chain. Things like shark, swordfish, king mackerel, many other kinds of fish that are big predatory fish. Big tuna, not the little ones, but the big ones, contain a fair amount of mercury. And where does this information come from? How did they decide this? Well, there were 2 studies, 2 very large studies involving more than 800 people in each, of 2 island populations that are exposed to mercury in their diets. In one, in the Faroe Islands. Now, the Faroe Islands is in the North Sea, roughly halfway between the north of Scotland and Iceland. And this is a shot of the Faroes. You notice there are no trees. It's too far north to have trees on it. It's a relatively mild climate for something that far north because it's right in the middle of the Gulf Stream, which keeps it warmer than it otherwise would be. And in the Faroes, they eat a lot of mercury. And basically, mercury appears to be a problem. Very many of the Faroese appear to be suffering from neural problems or subtle problems. They're not really bad, otherwise they'd notice it and do something about it. But they appear to be affected by the mercury they're eating. There's a second study, a similar number of people and done in a similar way, and lots of the same parameters were measured, in the Seychelles. The Seychelles is in the Indian Ocean, and by comparison, it's a tropical paradise. Mercury appears not to be a problem in the Seychelles, which is quite interesting. They have similar amounts of mercury in their diet, so they're different. What are the differences? In fact, what did the EPA and the FDA do? Well, they chose the mercury study of the Faroes as a guideline. And they chose the safe option. It's clear to us from our previous discussion that molecular form matters. And many factors could contribute. Maybe there's a difference in genetic factors. These are different kinds of people. The people who live in the Faroes are all Caucasians, whereas dark-skinned people live in the Seychelles. So there may be some sort of genetic factor. We don't know. There could be a difference in their diet, and in fact there is. Maybe different molecular forms are actually ingested, or maybe there's something else that's important, and we'll come back to this in a minute. In the Faroes, they eat quite a bit actually of whale meat. There's a sort of very controversial whale cull that happens several times a year in the Faroes, where large numbers of pilot whales are killed, and they eat a substantial portion of what they kill. And so they eat a lot of whale meat, whereas in the Seychelles, mainly fish. Now, if that's important, then it has impact for us because we don't eat much whale meat. Most of the world doesn't eat whale meat these days. This is just a cartoon I found somewhere on the web. It's well known that methylmercury in fish is a problem, but that's only half the story, it turns out. Remember that molecules are important. Methylmercury what? What is it? This is what you might call methylmercury, and this is an entity that doesn't exist. It's an impossible molecular fragment. It might exist in a gas phase with a positive charge on the mercury, but it certainly doesn't exist in our bodies, it doesn't exist in animals, it doesn't exist anywhere in our environment, or fish for that matter. Methylmercury is half the story. It's really not known what else is there because we've had to rely on conventional analysis to understand what's in the fish. And many possibilities exist, and these are just some of them. These are just some of the possible molecules that are reasonable to be occurring in a biological system, and I could draw thousands of them. There are many, many combinations in which mercury could exist, or methylmercury could exist in a molecular form. And we simply, until recently, didn't know what it was. Whoops, I went past something then. So we really need to know, this is a nice piece of swordfish steak, which incidentally I no longer eat. We really need to know what the molecular nature of mercury is in fish if we want to know whether we're going to be worried about it or not. We've got to understand the molecule. There's no good just knowing there's methylmercury something in it. It's very important to know methylmercury what. These are the only actual spectra or data that I'm going to show you, and I'm show them to you just to give you a flavor of what we look at day in, day out. This is the little dots here, you can see the best perhaps here or here, show what I call a spectrum, and what's important are the little wiggles and bumps and where they are. And they're matched against the solid lines, which are like standard compounds. They're reference compounds. And what we do, the simplest kind of analysis we do, by no means the only kind, is fingerprint them. So we'll look at our X-ray absorption spectra, that's what this is called, and we'll match them against a large number, maybe 30, maybe more, of reference compounds that are synthetic, chemically characterized small molecules that we really know the structure of already. So molecules that we know against the unknown. And you can see the top one matches. And so here's that methylmercury fragment, and it's revealed to us what the rest is. We now know that mercury in fish is a methylmercury cysteine species. We know that. Amongst all the possibilities, we can underline this one and say this is what it is. And this was published in Science of last year, about a year ago now, actually. Now we know that, we can move forward a little bit. Methylmercury cysteine, a long name, turns out to be much less toxic to fish. Now, other animals, mammals for instance, haven't been investigated, so we don't know about how toxic it is to mammals. But it's much less toxic to fish, little zebrafish larvae, which is a little tiny fish, freshwater fish, than some other methylmercury molecules. I mean, obviously, no, they haven't tested all other methylmercury molecules, but this is a strikingly less toxic form of methylmercury than many other species, and in particular, the other species that have been used in clinical experiments that have involved mice and things to determine how toxic the thing is. But we don't know about its toxicity in mammals. But what we suggested was, are the recommended safe levels for fish consumption lower than they need to be? We didn't say that they were, because that would be a bit rash, because we actually don't know. But there's cause to question it, I think. Now that we know the molecular form, we can actually move forward. And when we started saying this kind of thing, all kinds of fuss happened. And we ended up in a lot of newspapers and on the television a lot. The one I liked the best was the BBC coverage. The BBC, and I have to do this from memory, I hope I get it right. You probably know that I'm English from the way I'm talking. And at the time I did all these experiments, or we did all these experiments, I was at Stanford, but I subsequently moved to Canada. And what the BBC said, something like, Canadian scientists report, and then they talk about what we discovered, but British experts caution. They didn't say who the experts were. But I thought that was the best quote. The one I liked the best was actually the New York Times, which had a very nice, balanced article on it. And I'm going to change gears a little bit now and deliver a message from our sponsor. This is a commercial break, if you like. I want to advertise the next lecture, The Runaway Universe by Roger Blanford, who's the director of the Kavli Institute for Particle Astrophysics and Cosmology. It sounds like a very interesting topic, cosmology. So I think we've had one on SLAC, one on synchrotron radiation, one on antimatter and matter, one on metals and molecules and death and things. And now we've got cosmology, which is really quite a nice, diverse thing. This is to encourage you to join SLAC collections. And here at this URL, you could find out more about upcoming lectures. And also, if you want, please sign up for a tour of SLAC. And you can find out more about those right on the top level webpage there. Now back to Mercury. As I've told you, methyl mercury molecules are neurotoxic. But there's just a huge amount we don't know. We don't know how they work. We haven't got a clue. Actually, we do. We have little clues, but we really are in the dark about the bulk of the mechanism of how mercury molecules cause damage to nervous systems. One of the most curious things is that they exhibit a latency, a delay between exposure and the onset of toxic effects. I'll say more about that in a little bit. It can be as long as 150 days in people, a really long time. This shows you what a creeping, insidious thing some of these mercury compounds are. You can be exposed to them and not know anything is wrong for ages and ages, and all of a sudden it'll hit you out of the blue. And this is best illustrated by the tragic case of Karen Wetterhahn. This is a picture of her. She was a professor of chemistry at Dartmouth College, and she was using a substance called dimethylmercury that I've already mentioned. What she was doing involved a small amount, a very small amount of the material in a little glass ampoule like this one here, and she was transferring, she got a colleague of hers to transfer, she got a colleague to cool the compound to minimize the vapor pressure and the possibility of it bursting, the ampoule bursting. The colleague cut a little nick in the top of it with a file. They cracked the ampoule open. She used a little glass dropper, like the ones shown here, to transfer a small amount into a glass sample tube for a particular kind of experiment. Just a few drops were used to put those in. And then the tube was sealed up and with a glass blowing torch and labeled. She labeled it. And this happened in, I think it was August the 14th, 1996. She used gloves when she was doing this whole thing. She wore latex gloves. She wore safety spectacles. Everything was done. The entire procedure was done in a fume hood. This is a device that chemists use a lot when they're dealing with hazardous materials. And there are strong extractor fans in the top of it, and it pulls the air through to minimize exposure to you. So you're wearing gloves, a lab coat, safety spectacles. You're taking all the recommended precautions. At the end of the procedure, she noticed that she'd spilled just a few drops of this stuff onto her gloved hands. So it was on the glove, not her skin. At that time, she removed the gloves. She actually made a note of it in her logbook. And after everything was clean and tidy, she went home to her husband and her children. 5 months later, she experienced difficulty walking and talking. She went to a neurologist. Less than a month later, she was in a coma. Another 4 months, she was dead. There was not a thing they could do for her. Despite extensive and heroic treatment efforts, they could do nothing for her. This is dangerous stuff. What's curious, and what the point that I'm trying to get across, is the latency. You get poisoned, you're okay for 5 months, and then something bad happens. We have no idea how this works. And to understand how it works, we'll need to understand the molecular forms of mercury that are involved and how mercury is being modified in the body. One interesting way, mercury and selenium are linked in quite a curious way in human metabolism. It's been known for a relatively long time that if you take, let's say I know I'm going to get a dose of mercury, and I have the two together, and I accidentally drink the mercury and then accidentally drink an equal dose of selenium. Whoops, what'd I do? Oh, no. Okay, that's okay. Mercury given at the same time, the toxic effects are cancelled. So it's like the classical antidote. I was reminded of that Indiana Jones movie where he has an antidote in a little tube and he goes all around chasing around there. He gets the antidote and drinks it and he's fine. Well, this would be the selenium, but he'd have to drink it pretty quickly before the mercury got him. But the toxic effects of the mercury are cancelled. Now, we know a lot about how this works. I'll tell you about that in a minute. More curious, if I say, well, okay, later, I know I'm going to have some mercury, so I'd better have a dose of selenium right now. So I take the selenium later, and then maybe 2 hours later, I have a dose of mercury. Well, the toxic effects of the mercury are not only not cancelled, but they're hugely magnified. So it's much more toxic. Something weird is happening. The molecular form of the selenium, and I'm deliberately just giving the elemental names. I'm not telling you what molecules are used here because it'll just be too much detail, I think. But the molecular form of selenium is being transformed into something in our bodies that's combining in the 2 hours in between, and then it gets to combine with the mercury that you ingest to produce something that's absolutely devastatingly deadly. We have not a clue. Well, we have some clues, but we have no firm proof. We don't really know how this works. This is a big mystery, and it's one of the major things we're trying to understand. We do, however, understand this a little bit. Never mind the details of this picture. The thing I'd like you to concentrate on is this thing. When you take mercury and selenium together, what happens is you form a little ball of mercury and selenium, like a little ball bearing, maybe 100 atoms in there, surrounded by other molecular fragments, never mind what they are. And this floats around in your bloodstream, is moved to the liver, and is stored there forever until you die. This is how it works. This is how the cancellation of the toxic effects is manifest. We understand this in some detail now, thanks to experiments done at SSRL. Quite a curious thing to think you might have these little ball-bearing things in your blood. I'm going to move on. I'm going to talk about arsenic. Arsenite and selenite are the names of two common molecules that contain arsenic and selenium. And they're about as toxic as each other, and in isolation by themselves, they're each lethal at elevated levels. Remember, we need selenite in our diet. We need selenium in our diet. It doesn't have to be selenite, but we need selenium in our diet. So actually, selenium is pretty important because the level between enough in our diet and the toxic level is actually not, the difference between those two levels isn't very great. In isolation, they're each lethal at elevated levels, and their toxicities, as I said, are approximately equal. When they're taken together, like the mercury, but the mechanism is quite different, when taken together, their toxic effects are surprisingly cancelled. Now, maybe it's not that surprising because mercury did it, but it works in an entirely different way. It's quite an old piece of knowledge. In 1938, it was first published that rats fed on 11 parts per million selenium in the form of selenized wheat. This is wheat contaminated with selenium. In 60 days, fed on only that amount of selenium, they were all dead. Every one of them died. But if they were fed not only on the selenized wheat, but given the arsenite in their drinking water, in this case they gave 5 ppm, they all survived. Now, it was later discovered that this was even more effective if you gave exactly equal doses of arsenic and selenium. And the the thing that emerged over the years was that a lethal dose of arsenite or selenite can be completely counteracted by an equal and otherwise lethal dose of the other one. So you could take 2 lethal doses together, you're fine, you don't die. Very surprising and very interesting. How does this work? Well, once again, synchrotron radiation, our X-rays revealed the species in bile. And this is excreted in the bile. What happens is the arsenic and the selenium come out together in this molecular entity. Now, this is the name we gave it. We called it the seleno-bis S-glutathione arsenium ion. Doesn't really matter. But the important bit is in the middle with the atoms that I've labeled. And you can sort of forget about the outside bits and just concentrate on the middle bit. Here we can see an arsenic atom shown sort of a shiny metal-looking purple atom bound to this golden-looking atom, which is selenium. There are 2 sulfurs, and they're incidental to this point. The point is that it's a 1-to-1 selenium complex. There's 1 selenium to 1 arsenic in this molecular form. And we were thinking about what was happening in Bangladesh. There still is a very major problem with low levels of arsenic in the drinking water in Bangladesh. It affects just an enormous number of people. Basically, the arsenic is coming from what they call tube wells, which are just simple artesian wells. Perhaps some of you here get your water from artesian wells. And there turns out to be, I won't go into the reasons how the arsenic got there, how long it's been there. That's shrouded in all kinds of debate. But it turns out there's a small amount, not a huge amount, of arsenic in the drinking water. But it affects 30 to 75 million people. That's just an awful lot of people. Neil told you I'd moved to Canada. 30 million people is the population of Canada. I'm English. 75 million people is more than the entire population of the United Kingdom. This is an enormous number of people that are affected by this problem. And this is not the only place in the world that we have these problems going on. There are problems in China and other places too. However, most interestingly, there are some locations in the world which have similar arsenic levels in the water, but actually have less severe health problems. And that was very curious. Why should one population be affected poorly while the other is, barely affected at all? And the mechanism of this low-level arsenic poisoning is really unknown. We don't know how it's doing it. So, what are the symptoms? Well, I won't go into lots of detail, but they involve a whole bunch of skin problems, malignant tumours, and then death. There's no real good information on how many Bangladeshis have died. And these are typical, I wondered whether to show you gory pictures, and these are some of the less gory ones. These are typical pictures of what the medics call arsenic lesions on the hands and feet of Bangladeshi sufferers. And you can see it looks pretty unpleasant. Now, as I've mentioned, selenium is an absolutely essential trace element. And deficiency results in skin problems, malignant tumours, and death. Sound familiar? We all take, actually many of us, probably many of the men anyway, I do, take selenium on a semi-regular basis. Now remember this molecule, for every arsenic that is removed by the formation of this molecule, 1 selenium is lost. One of the essential seleniums in our bodies is lost. So our hypothesis that we published was, does the low-level arsenic poisoning in Bangladesh actually cause selenium deficiency? We still don't know for sure. You can't do experiments on human beings. But it's striking that the symptoms of selenium deficiency and low-level arsenic poisoning are indeed very similar. And there are other parts of the world, but the Bangladesh and the other regions in the world that are affected by this so-called chronic, which means sort of long ongoing, low-level arsenic poisoning, are actually low in dietary selenium. So they don't have much selenium in their diets anyway. So perhaps this is an explanation of what's going on. We hope so. It turns out that there's a Chinese group who's studying one of the affected areas in China and administering selenium supplements. And indeed, in that case, it does appear to have quite striking beneficial effects. So synchrotron radiation perhaps allowed us to help quite a lot of people. We hope in due course that will come true. I'm going to say a few words about what's next, because I've told you a lot of problems. What do we want to do next? Well, many of the things we want to understand are to do with mercury. Arsenic is close to being solved now, at least from our molecular point of view. We want to understand the methyl-mercury latency. We want to understand the detailed nature of mercury-selenium interactions. And we want to push detection limits to even lower levels. One thing I haven't mentioned is what has made all these experiments possible are really the super bright X-ray beams at facilities like the Stanford Synchrotron Radiation Laboratory at SLAC and other places in the world. But all of these experiments were actually done at SSRL. And the new detectors, highly stable, highly automated experimental facilities here, basically have allowed us to push detection limits to really low levels, levels that people really can't achieve elsewhere. When you publish a paper in a scientific journal like Science, you get reviewed by other scientists. And one of my reviewers wanted to know why I was able to do experiments with 1,000th of the sample that he could. And the reason is that here at SLAC, they're very good at doing what they do. So we want to push detection limits to even lower levels. And we want to look at something called custom chelators, which I believe at least is the promise of the future. And I'm going to talk a little about those. What are these custom chelators? Once we know the molecular form that's in our bodies, we're in great shape to design a way to get it out. So what we've been doing is we've been using some fancy computer techniques to design specific candidate drugs. They're not drugs until they're actually really ready to use, for treatment of poisoning. And one way you can view this is a custom chelator works like a lock and a key. It's very key specific. The metal is the key. The chelator is the lock, for example. They only go together with the right lock and key. Other metals really won't fit as well as the target metal. So this is a molecule that's tailored to fit the particular thing you're trying to grab and remove and make it unavailable for poisoning in the body. And the goal, of course, is to remove the metal from the body. And this is one candidate molecule, and you can see it's grabbed a mercury atom here. There are 3 sulfur atoms that are positioned in the absolutely ideal places for binding. And I should say this is a molecule that doesn't actually exist yet. It only exists in the computer. So it's calculated, and we understand a lot of its properties from what the computer can tell us. But it doesn't really exist yet, but it probably will. So I'd like to summarize. Molecular form matters. It's really important to know the details. There's absolutely no use understanding how much lead, mercury, whatever it is, whatever the metal, arsenic, you really have to know what the molecular form is you're dealing with. You need to understand the molecules, not just the levels of the metal. Molecules are very important. They can make all the difference. And we must, in our research, examine this and not just the levels. It's traditional, especially in medical research, people have used methylmercury compounds without even saying what the methylmercury compound is. And there are published papers in the literature, many of them, that say methylmercury was added and that we don't know what the methylmercury thing they added was. They ignore the rest of the molecules. It's very important. We must understand the molecules. And synchrotron radiation hopefully provides a key promise of better health for all of us. Now, I'm going to acknowledge the Stanford Synchrotron Radiation Laboratory. The facilities here are really outstanding, and without the benefit of those, none of this could have been accomplished. And what we do is a very small part of the overall research at SSRL, which is supported by the Department of Energy and the NIH. And these are all the people who've given me grants. And lastly, I'd like to thank you for your attention. I think we have time for some questions.
With X-ray, is it possible really to distinguish the molecular bonds in the molecule, or with X-ray to see that mercury is present there?
Well, with the technique that we call X-ray absorption spectroscopy, we can tell what the neighboring atoms are in a molecule. Ordinary X-rays, like your doctor's office X-ray, won't do that. It'll just tell you there's something that's dense to X-rays there.
How do you isolate the compound that you want to
Well, the compound, we don't need to get a single molecule. We can take a whole piece of tissue. People can put, you know, a finger in the beam. They'd be a bit foolish to do so. Probably, actually, they wouldn't be allowed back in the facility if they put a finger in the beam. In principle, you could put a piece of you know, a biopsy in the X-ray beam and look at it directly. You wouldn't damage the tissue, not significantly anyway, in the time that you'd need to measure it.
Isolate the people you're looking for a specific compound?
Well,
is that the reference frame?
you get what's called a spectrum. What you do is you vary the energy of the X-ray and you look at the different ways in which it's absorbed. And I don't really want to go in the details here because it's kind of complex, and I think it would bore most people. So if you want to know more, I'll tell you afterwards. Come down and talk to me.
Is NMR useful?
Mercury 199 NMR is a useful probe. In fact, that's what Karen Wetterhahn was doing when she was poisoned. Unfortunately, its sensitivity is way too low to actually measure real samples. So you couldn't put fish into an NMR machine and see anything. Our sensitivity is perhaps 1,000,000 times greater than NMR.
Given the studies of sheep with the mercury fillings in their teeth, why haven't you had your fillings out?
Oh, the simple reason is if you have your fillings pulled, that's a great question actually, it's something I probably should have said. If you have your fillings removed, you get a way bigger dose of mercury than you're going to get if you leave them in. Because the action of pulling them out would be much worse than leaving them in place. So mine are staying put. Why? Well, because always some is lost and always some is ingested. You know, that despite how, you know, dentists are very careful with mercury these days. Many of them put on a thing they call a dam, which sort of fits around your tooth and prevents any little drops getting lost in your mouth and swallowed. But inevitably, there's always some ingestion. So it's better if they stay put. I think you were first.
I just went to the dentist and I was actually talking with him about this. He said the mercury that's still in the cavities that they have is sealed with inside. So there's always there's a little risk, but it's not like it's just it's not. Oh shoot.
Well, the mercury
it's not just open-faced in your mouth. Or,
if you look
yes.
in the mirror and you see a grey metallic-looking substance in your mouth, that is silver mercury amalgam. It's in your mouth, but it's not very available. Not much of the mercury comes out, so I wouldn't worry too much about it. I'm not very worried. I've got loads of fillings. Go ahead.
Do the same problems that you've been describing with mercury and arsenic exist with lead, in that the molecular form that lead is in?
Yes. The molecular form of lead will be important. So same things apply. Go ahead.
You only mentioned selenium, but other things like, for instance, thiamine and methionine and some of the other chemicals are also kind of like combining with mercury, and then they also can make something else, which then makes a person possibly thiamine or methionine deficient.
I don't know of anyone becoming methionine deficient. Methionine is a very common amino acid in all the protein we eat.
There's a Russian publication, it's by Trachtenberg, and he has written quite a bit. This was written in 1974.
Are you sure it's methionine? Because I think.
Methionine, I'm positive.
Okay. I don't know that works. Sorry.
Well, methionine has a sulfur component, and sulfur is very attractive.
Not in methionine because it's a thioether, so it will not form very strong covalent bonds like thiols will. So it won't.
But apparently, in those studies that they did on animals, it made quite a bit of a difference.
Okay. Well, I'll look that up. Thank you.
I wanted to share a counter opinion on the mercury in fillings. Just as my understanding, mercury in a filling is not it's not a molecule, it's an amalgam, it's an alloy. So it really is mercury with silver that are intermingled. And I've read some research that suggested that the mercury that gets into your mouth in concert with the organisms that grow in your mouth, it may be becoming more bioactive. So, elemental mercury may not be that dangerous, but there's not a lot of evidence that it's staying elemental mercury.
I can comment on this.
Okay.
Okay. Yes, you're right. It's a metal, but I'm trying to give a general public lecture here. I have to generalize a lot. Metallic mercury is possibly a hazard to us if you ingest it. Now, some of you may know that metallic mercury was used in the medical industry years ago to cure constipation. The technique involved taking a draft of metallic mercury, sitting on a commode, and the blockage was cleared. So it's not as bad as all that. If people could do that and survive with no apparent ill effects, then it can't be that as deadly as all that. But it is true that there's a lot of concern these days about a type of bacteria in our guts called sulfate reducers. Perhaps at any given time, 50%, as many as 50% of us might have sulfate reducers in our guts. What sulfate reducers can do is they can take the mercuric mercury, or the forms of mercury that are inorganic salts, and elemental mercury, and they can turn them into the neurotoxic methylmercury species that we know are deadly. So it's recently been found that these sulfate reducers can do that, and whether they actually do that in our guts, we don't know. That's an unknown. And in fact, it's one of the questions we could answer if we chose. Yes?
What about the mercury content of wild salmon?
Wild salmon. Wild salmon aren't particularly high in mercury compared to things like swordfish and tuna. I don't know how much we should worry about eating fish, because as you've heard, the data that the guidelines are based on is a bit ambiguous. I'm perfectly content to eat wild salmon myself, but I've stopped eating swordfish and shark, which are higher. Yeah, go ahead.
Yeah, well, that actually was my second question, but my first question was, do you have any information as to how much of the methylmercury that is in fish has been human introduced in the last century or so, and how much of it is naturally occurring?
Okay, I don't have direct information on that, but my understanding is that mercury has been in the environment for very many years. Undoubtedly, some of it's from our activities, from human beings introducing mercury into the environment, but most of it was there all along. It's been there a long, long time. Go ahead. Oh, we got one here. Okay.
It looked like your spectrum showed not only methylmercury cysteine, but also methylmercury chloride?
No, there's no methylmercury chloride there.
Okay.
No, there wasn't. It was there as a possible compound, yeah. Okay. Yeah, we didn't find it there. Those were like things we checked and examples.
It wasn't clear which was the data and which was the method.
The little dots were the data.
Why is there higher reported mercury in farm fish?
There isn't. There's lower. The concern with farm fish is to do with PCBs and other toxins. There's lower. If you go for farmed, we always, when we run a piece of fish, we always tell which is the farm one. The vendor might say, "Oh, this is wild caught," which of course is more expensive. Well, no. The mercury is real low in the farm one. Yeah, go ahead.
You spoke about the connection between rapid onset autism and vaccines. Could you elaborate on that?
Gosh, well, okay, this is a very touchy subject, and I didn't want to really elaborate on it too much. But a few years ago, it was suggested that thimerosal, which is a compound, I'll give you the name of it, it's ethyl mercury thiosalicylate. It's a compound that was introduced in vaccines in the 1930s. It's been there a long time. And it's introduced as a bactericide and as a fungicide. So they put it in little bottles of vaccine that many needles go into. Something like your flu shots have about 0.01% by weight thimerosal in there. So many needles go into that big bottle, and they want something in there to prevent any accidental infections that might be introduced. It should be sterile, but of course it's never perfect, right? From growing, you don't want to introduce a culture of some miscellaneous bacterium or fungi into someone's bloodstream. That would be disastrous. So they put this stuff in there to stop bacteria and fungi growing in vaccines. The controversy is all about for use in childhood vaccines. Now, it's been discontinued in this country, as I mentioned. It's been discontinued in many civilized countries. In fact, 2 weeks ago, I was in the paper in the United Kingdom because it had just been discontinued in the United Kingdom. And so the Daily Telegraph and maybe a couple of others carried this story, and it's a big deal. Now, they say the medical establishment is firmly of the opinion that, or the bulk of the medical establishment, that thimerosal is not a problem. Here's what I think. I think it's crazy stuff to have in a vaccine. It was introduced in the 1930s. We know it's neurotoxic. Why on earth are we putting a neurotoxic thing in a vaccine to begin with? And this is an archaic molecule that would never get approved these days. That's my personal opinion, and I'm going a bit out on a limb. I don't know whether it causes autism. I don't know whether it causes ill effects in anybody, and I don't believe anyone else does for sure either. There was a recent study from Cornell that showed that some of the symptoms of autism occurred when mice of a particular strain, and it was a strain-dependent thing, were given thimerosal. And so strange things happen with autism, like there's this social isolation. Also, autistic children's brains tend to be a little larger. The mice had larger brains. They showed isolation-like symptoms. So this study was perhaps the first and most compelling evidence that I know of. It's published in a journal called Molecular Psychiatry. But there is no firm tie between mercury and autism. I think we might as well get rid of it. If we understood how mercury was bound in the brain, we might be able to design a custom chelator to get rid of it. That's kind of far-fetched, but you never know. Steve?
Whenever I got cut, my father would go to the medicine cabinet and take out a red bottle of Mercurochrome. Should I be worried because it's dangerous?
I think it's too late for you. I don't know. Go ahead.
You said that men want more selenium in their diet, like you take selenium supplement pills.
Yeah. Why? Selenium has been linked with, is linked in a good way with the incidence of prostate cancer in men. Basically, a study which used selenized yeast, which is yeast that has been grown up in a background of selenium. Why do they use selenized yeast? It's because they could smell the selenium compounds in the placebo, the difference between the placebos when pure compounds were used, but when they used something a bit stinky like yeast, then they couldn't smell it. So they used this yeast and they used regular yeast and they gave them little pills made of the selenized yeast, and there was an unexpected and a really striking beneficial effect. The incidence of prostate cancer was really reduced when in the group that had the selenium, and since then people all over the place have been taking selenium to try and, it's advised. And it's a good idea to take selenium, but I would caution you that not every selenium supplement contains the same thing. We've looked at bottles of selenium supplements on the shelves from health food stores. Even the same vendors with different batches, they have remarkably different compositions. So who knows whether they're helping or not.
Just a point. If you're giving, the study was done with yeast.
Yes.
Selenized yeast.
Yes.
And you know that there's a group. Do we know how yeast changes the intestinal flora to get your sulfur reducing or something?
Well, no. The yeast will probably be killed by the acids in the stomach. It won't change the intestinal flora. This is just ordinary baker's yeast. It's not going to survive your stomach. Whoops, up there, sorry. Well, you might try a different form. One form you could try is take a Brazil nut every day. Now, Brazil nuts come in two varieties, those with shells on, those without shells on. Those with shells on, all Brazil nuts are harvested from the wild, And we know what they contain. They contain a form of selenium called selenium methionine, and it's very pure. It's just selenium methionine. And there's an awful lot of it. Now, the ones with shells on, they're harvested from the wild, from trees, and the Brazil nut trees are about 50 feet high, and the Brazil nuts are packed in like little segments into this great big grapefruit-sized thing, this fruit that falls. It would probably kill you if it landed on your head. But they go and they pick these things up, and they crack them open. And in the parts of Brazil that actually have the technology to remove the shells, there's no selenium or negligible selenium in the soil. And so those shelled Brazil nuts don't contain much selenium. But if you have the ones with the shells on, they come from a different part of Brazil, usually. And it's a good job they don't have the technology to remove the shells, because if you could eat a lot of them, like you would do, perhaps if you didn't have to crack that shell off, it's difficult to get a Brazil nut shell off, then you'd probably get poisoned. So it's like a self-regulate. So try one shelled Brazil nut a day. You might find... I bet it's good for, yes, it's good for all of us. All of us need it.
What about
yes.
pumpkin seeds? Don't they have selenium?
I would imagine it would depend where the pumpkin is grown. I don't know anything about the form of selenium in pumpkin seeds, but I bet you, your garden might be quite different than Half Moon Bay, unless it's in Half Moon Bay. Go ahead.
It's my understanding that mercury will adhere to organs. How would you test if you suspected some sort of mercury poisoning?
Adhere to organs. I don't know about that. What it will do is it will get into your bloodstream and it will involve itself in your metabolism. Not much is known about how its molecular form inside us, in our nervous systems. Not much is known, and the experiments that need to be done are to take pieces of tissue and put them in the X-ray beam and understand them. And what we're going to be doing, I hope, is get pieces of people, biopsy samples of people who naturally have a lot of mercury in their bodies, like from the Seychelles. So I hope we'll be able to do those experiments in due course and hopefully understand what's going on. And also samples of brain from people who've died in road accidents and things like that. So hopefully that'll come to pass. Yeah?
Would Brazil nuts be beneficial to the prostate, and if so,
Yes.
where would you put them?
Well, you would put in your mouth, chew them up and swallow them. The curious latency, yes? Mm-hmm.
If one could do an analysis of neurotoxic effects at the time one was infected, couldn't one work backwards in animal studies? And I guess a corollary to that is this exposure so rare that it wouldn't be worth doing the trouble?
Well, I think it's worth understanding because it'll give us a clue as to how methylmercury poisons us, if it is poisoning us, when we eat too much methylmercury in fish. It's certainly worth studying because, I mean, most of us here have probably eaten fish at one time or another. I love fish. I eat maybe 2 meals a week fish. In the Seychelles, they eat fish roughly 2 meals a day, so they eat rather more fish than I do. Yes, you need we will need to expand the study to try and understand where it goes. There's a lot to do. What I did mention right at the end is this is only something that's recently been doable. Right? We haven't always been able to do these experiments. We basically got a 3-fold or a 1,000-fold increase. I was going to say 3 order of magnitude. That's a 1,000-fold increase in signal to noise in the few years we've been doing these experiments. Go ahead.
Are you aware of people investigating the commercial applications of things like selenized wheat or chelators?
There are certainly a lot of commercialization of selenized yeast. Tons and tons of different health food companies make the stuff, but they seem to vary widely in what's in them. Most of the selenium in many of the samples we've looked at, I'm not going to name names of products, but most of it tends to be elemental selenium, alpha elemental selenium, which is the red form. And that's biounavailable. It'll pass straight through you without doing you a jot of good. I reckon you should eat Brazil nuts. That's what I reckon.
Excuse me. One of the main reasons why I come to these is, in fact, that we have free Coca-Cola and cookies outside. I'm getting kind of restless to go out there and get them. Could I, in Brazil nuts too, which I'm going to try and eat and introduce as many as I can. Graham, I'm sure, will be delighted to continue answering questions, and in fact we have other scientists who are out there. You can recognize them because they have their names stuck onto their chests. And so if you'd like to continue, please go outside. We can have a soft drink and some cookies. But before that, let's thank Graham again for a fantastic talk.